Thank you very much, Andrew, and thanks also to Natalia for inviting me to talk at this wonderful workshop. So in my talk, I'm going to pick up on a topic which has already featured a number of times during the meeting, and that's the idea that specific factors, for example, dietary factors or obesity in one generation could influence health outcomes in subsequent generations. And I will focus on a specific mechanism, epigenetics, which could potentially mediate these intergenerational signals. So many people here, of course, are familiar, that in certain cases, familiar with this concept. For example, epidemiological studies show us that poor maternal nutrition is associated with adverse outcomes in offspring. But the mechanisms are often poorly understood, and of course that makes it difficult to identify the specific nutrients, for example, which might be driving these adverse outcomes. And as we all know, these mechanisms are very hard to study in humans, particularly with observational data, because there are many potential confounders, and the causal pathways are interlocking and often very hard to elucidate. And that's one reason why in my field, in nutritional epigenetics, some of the most interesting observations have come from so-called natural experiments. And here's one example, the Dutch hunger winter series of studies. So the Dutch hunger winter is a period in the Second World War when a population, geographically restricted population in the Netherlands, was subject to a blockade which resulted in an extreme famine, but over a very short window of time, just a few months. And in these studies, individuals who were exposed to these famine conditions during gestation were compared to their unexposed siblings. And here's just a few examples of some of the findings from these series of studies. So prenatal famine has been associated with increased incidence of obesity. So this is in adults in much later life. Also dyslipidemia, increased incidence of diabetes and schizophrenia. And one interesting observation from some of these studies is that the period in early gestation, the first trimester of pregnancy, appears to be a particularly sensitive window for these particular effects, these types of effects. This is also revealed in another much more recent study, here looking at the effect of fasting in Ramadan on under 5 mortality. And here, once again, you can see that it's the period around the first trimester, in fact, around conception, where we see much increased incidence of under 5 child mortality. So again, pointing to this sensitive window in early gestation for the transmission of these effects across generations from mother to child. Moving on to some other natural experiments, so there's another very interesting series of studies in the Överkalix cohort in Sweden. So here they looked at the grandchildren of grandparents who were exposed to large fluctuations in harvest quality back in the 19th century, and they looked at the effects in grandchildren. And what they found here was quite complex, but what they found was that grandfathers' exposure to poor harvests in the prepubertal period predicted increased mortality in their grandsons, whereas grandmothers who were exposed in the in utero period, during pregnancy, predicted their granddaughters' mortality. And this first observation has now been replicated very recently in a very large cohort of over 10,000 grandchildren, another Swedish study. And in fact, there are now quite a number of these natural experiments, all with diverse outcomes and some heterogeneity in effects. But I think you can broadly summarize them as suggesting that exposure to prenatal adversity is associated with a whole range of adverse outcomes of the offspring in later life. And as many of you, or some of you will know, my work is in the Gambia along with Andrew Prentice. And I think we have our own natural experiment here in the Gambia, which thankfully involves exposures, much less severe exposure than some of the examples I've just highlighted. And our exposure is one of seasonality. So here is our rural research station in rural Gambia in the village of Keneba, and this is Keneba in the dry season. And in contrast, here is the same aerial shot, this time of Keneba in the rainy season. And what we find in the Gambia is there are differences in diets across seasons, there are differences in the burden of infections across seasons, and also in energy balance, for example, which is manifested in weight changes between seasons in pregnant women, for example. So this gives us our own, what we like to call an experiment of nature, a sort of quasi-randomized controlled trial whereby births and conceptions are randomized to one season or another. And probably one of the starkest effects that we've seen in terms of seasonality is an effect on season of birth. So this is an analysis which was carried out by Sophie Moore nearly 20 years ago now. And what she found was that individuals born in the rainy season are significantly more likely to die prematurely than those born in the dry season. Now, thankfully, these are rather low numbers of deaths that are actually represented in this plot, but it's a highly significant effect nonetheless. And notice one of the interesting aspects of this is that these survival curves only start to diverge around the time of late adolescence, suggesting that there is some sort of memory of an earlier life adverse exposure being carried forwards into later adolescence and beyond. And it's observations like these which have really sparked our interest in epigenetic modifications as a potential mediating mechanism for this, for reasons which I hope will become apparent as the talk progresses. More recently, we've stepped back a generation and looked at the effect of parental season of birth on offspring anthropometry in this case. And what we found here is that a mother's season of birth predicts her offspring birth length, whereas a father's season of birth predicts his offspring's height at age 2 years. So this paternal effect is particularly interesting because it's suggesting that the effect of an early exposure in the father's life is affecting his son, which the most parsimonious understanding would be that this must be mediated in some way via the father's sperm. So for those who aren't familiar with epigenetics, I'll just give a brief introduction to the concept. So epigenetic modifications are heritable changes to the genome that can affect the degree to which our genes are expressed, but without altering the underlying DNA sequence. And I think there's rather a nice analogy here in terms of musical notation. So you can think of the epigenetic marks in the genome a bit like the dynamic marks you might see in a musical score, which tell the musician whether to play a particular section loudly or softly. So we can think of the musical notes in this analogy as the genetic code, and the epigenetic marks are the marks on top of the genome which are determining the degree to which specific genes are being expressed or underexpressed in different cell types and different cells. So why are we particularly interested in epigenetic modifications as mediators of these early life exposures and impacting on later life health outcomes? Well, this is because in animal models and in some human studies, it's become apparent that these epigenetic modifications are particularly sensitive to the early environment. Also, they're generally stable throughout the life course, and as I've said, they influence gene expression. They've also been associated with numerous diseases and traits, and there's also this potential for transmission of epigenetic signals across generations. So But what about links to human health in terms of epigenetic mechanisms? So one particularly nice example comes from genomic imprinting. So there are a number of genes in the human genome which are imprinted. So regular genes across the gene and the vast majority of genes are expressed equally, depending on, irrespective of whether the copy of the gene comes from your mother or your father. But at imprinted genes, we find that only one copy of the gene, either the paternal copy or the maternal copy, is expressed. And this differential expression pattern is determined by methylation marks close to the gene, which determine which particular allele, whether it's the maternal allele or the paternal allele, is expressed. And there's an interesting hypothesis on why this has evolved in mammals, and it's to do, we think, to do with a sort of struggle between the mother and the father, a sort of tug of war between the mother and father in terms of constraining or promoting fetal growth. And when you look at the genes involved, some of the imprinted genes, this is borne out. So some of the paternally expressed genes promote fetal growth, whereas many of the maternally expressed genes suppress fetal growth. And when this mechanism goes wrong, it leads to several severe imprinting disorders, some examples here. And as you can see, many of these do involve severe dysregulation of fetal growth as well as other outcomes. But there are also more subtle effects which have now been revealed, small changes in methylation at imprinted genes, which can also influence postnatal growth and birth weight, for example, some of which have been linked to certain exposures, including nutritional exposures. Another nice example, very recent example, is a discovery of a DNA methylation signature in every cell of our body, which is very strongly correlated with our chronological age. And accelerated epigenetic aging, when we look at this signature, is very strongly associated, predicts adult mortality. So you can see here in this example, if you measure epigenetic age, in this example at age 50, those individuals who show an accelerated epigenetic age 10 years in advance, which is, you might suppose, as a sort of biomarker, a biomarker of accelerated biological aging. If you compare those with a 10-year epigenetic accelerated age to those with a decelerated, a younger epigenetic age by 10 years, there's a 5-year difference in predicted time of death. So that's another example of an epigenetic signature which is associated with mortality. So I've mentioned that the period in early gestation is particularly sensitive to changes, to environmental exposures. And you can see from this plot why that would be. So that the period in early gestation is a particularly sensitive period where there's widespread reprogramming of the epigenome. So here we've got the example of DNA methylation, which is one epigenetic mark. And you can see that just around conception, in the first few days following conception, in the first few cell divisions following conception, the methylation marks on the sperm and the oocytes are largely erased. And that's necessary in order to render the embryonic stem cells in a pluripotent state ready to acquire their cell-specific marks after implantation of the blastocyst and uterine wall through gastrulation and beyond, when the cells acquire their somatic cell-specific marks. And this occurs, as I've mentioned earlier, across the epigenome, apart from at imprinted genes in a few other locations, but imprinted genes specifically where the parental specific marks on either the sperm or the oocyte are maintained across this, so they escape this erasure in early gestation. There's then a second wave of demethylation, the primordial germ cells. Before these cells then go on to acquire their sex-specific marks in the case of males later in gestation and with females postnatally. So you can see here that there are these windows of sensitivity potentially where environment might play a role in disrupting these processes or altering the trajectory of these processes. And for those of you who know our Gambian work, we've been particularly focused on the periconceptional period in this respect. So as you might imagine, this has important implications for how these marks might be transmitted across generations in so-called epigenetic inheritance. And when we think of this, it's very helpful to, I think, separate two cases: inheritance down the male line and inheritance down the female line. So beginning with the female line, if we imagine a pregnant mother who's exposed, who has a particular exposure, then her offspring will be directly exposed in the womb, but so will her grandchildren, because of the developing primordial germ cells that are being carried by her offspring in gestation. And it's only the third generation in this case which won't be exposed. So if we're talking about the transmission of epigenetic signals across these transitions, as I described in the previous plot, then it's only in this third generation where we see true epigenetic transmission not resulting from an initial exposure in the pregnant mother's generation. And for this reason, we use different terminology to describe these two phenomena in terms of epigenetic inheritance. So the epigenetic inheritance as a result of direct exposure is termed intergenerational inheritance, whereas in the third generation, in the maternal line, we call it transgenerational epigenetic inheritance. And Ranjan gave this example in his talk yesterday with a rather nicer picture than the one I'm showing here, which is just some Windows clipart or something. But what you can see here is if the mother is exposed while she's pregnant, the potentially three generations are directly exposed: the mother, the offspring, and the grandchildren. So switching now to inheritance through the male germline. So here, it's only if the father is exposed, it's only his offspring that are directly exposed, and his grandchildren are not directly exposed. So in this case, we have intergenerational inheritance in the first generation and transgenerational epigenetic inheritance potentially in the second generation. And if you think back to the over-Karlik study, which I mentioned earlier on, so this is a potential example, therefore, of transgenerational epigenetic inheritance because we have exposure in the grandfather and an effect being manifested in the grandson. Now, there's a great deal of interest in the potential for transgenerational epigenetic inheritance to occur in humans. But if you talk to any epigeneticists, they will be highly skeptical of this effect occurring in humans in all but very, very rare cases, if at all. And that's because of this dual waves of epigenetic erasure, epigenetic marks at conception and in the primordial germ cells. So for true transgenerational epigenetic inheritance to occur in humans, they would have to evade these two waves of erasure. And the evidence is just not there yet in humans. We have the epidemiological evidence of effects manifesting across two or more generations, but not the epigenetic evidence. Where we do have strong evidence is in plants. So here's an example from maize. So in this example, the color differences are caused by epigenetic changes. And these are propagated over multiple generations, in this case over 10 generations. And here, the epigenetic mechanism is RNA-based. And here's an example from animals, this time in the nematode worm. So here, worms are exposed to elevated temperatures of 25 degrees C over 5 generations in this experiment. And what you can see is the worm there glowing red, and that's because this has an effect on a heat shock protein, which is being labeled with a luminous reporter, so the worm glows red when it's showing increased expression of this gene in response to the elevated temperature. And then in this study, what they did is they followed up over multiple generations, this time removing the exposure, and they showed that the effect persists for up to 14 generations in the absence of the exposure. And here, the mechanism is another epigenetic mark, this time modifications to the histones scaffold of DNA. So as I said, the evidence in mammals, and in humans in particular, but in mammals then is less clear. But here's one example which has really attracted a lot of attention and again, a lot of controversy. But I think this is particularly striking. So here we have a mouse, this is the F0 generation, exposed to an odor, acetophenone, which apparently has a cherry smell. And at the same time that the mouse was exposed to this odor, it was given an electric shock. And it was then, so there was a conditioned fear response. The mouse was then conditioned to fear the exposure to this odor, this specific odor only. Sperm from this mouse, this is a male mouse, were then taken, and the next generation was conceived through IVF with an unexposed female, and this fear response was still apparent in the second generation. And they then continued this into the third generation, into the grand progeny, and again the fear response is still apparent. So this is a potential example of transgenerational epigenetic inheritance, because down the male line, it's persisting across these waves of demethylation across two conceptions just through the sperm. And in fact, the experimenters found there were changes in the sperm methylome in both the F0 and F1 generations at the specific receptor gene for this particular odor. And indeed, there were neuroanatomical changes in the particular pathways receptive to this odor. And interestingly, these effects could be reversed if the F0 generation was conditioned back to not having a fear response in response to the odor. And I think that's an important observation, because it's suggesting, first of all, the marks can be erased, but also suggesting that it might be the phenotype in the F1 generation which is sparking the changes in the F2 generation, and not necessarily directly this idea of epigenetic inheritance across two generations. So if this is to occur, as I've mentioned, we'd need to see evidence of areas in the gametic methylomes that can evade these two waves of erasure. And there is evidence actually in humans that there are parts of the epigenome which do escape these waves of erasure. And the majority of them are associated with transposable elements in the DNA. These are evolutionary, very young elements in the DNA, which we're also very interested in our Gambian studies, because lots of the loci that we find affected are also associated with these elements, opening up at least the possibility that these marks could be transmitted across generations. There are other potential mechanisms as well, a lot of interest in the possibility of carriage of RNAs in sperm in terms of mediating these transgenerational signals. And here's an example of this in a recent mouse study. So this is a mouse model where the fathers are exposed to severe postnatal stress, which leads to metabolic dysfunction and depression in his offspring. And in a very elegant study, they've taken the sperm from these fathers, extracted specific sperm RNAs, and then injected these in zygotes from unexposed pairings. And they do indeed find that the phenotype is recapitulated in the offspring's generation, mediated via the sperm RNAs. Right, you may be pleased to hear that I'll switch away from the topic of severely stressed mice and switch back to humans. So we know there are a vast array of exposures, including nutritional exposures, which have been associated with epigenetic changes in humans. And we have a particular interest in nutrition, of course. So how does nutrition influence DNA methylation? Well, Ranjan has already touched on this yesterday. It's through the action of these interlocking pathways, which are collectively known as one-carbon metabolism. And these are responsible for providing the methyl groups, which are required for the methylation of DNA, and also many other methylation reactions in the body. And these pathways have many nutritional inputs that we get from our diets. So various B vitamins, folate, riboflavin, and other dietary sources, betaine, choline, and so on. And switching back to our Gambian experiments, we find that many of these so-called methyl donor nutrients vary significantly by season in the Gambia. So, for example, you can see, so these are biomarkers, methyl donor biomarkers, measured in pregnant mothers' blood plasma. And we see, for example, increased levels of folate and riboflavin in the rainy season, increased levels of homocysteine and dimethylglycine in the dry season. Okay, I lied about the mice. There's one more mouse slide here. So really, the strongest evidence in mammals for how early nutrition can influence later health outcomes and phenotypes mediated by epigenetic mechanism comes from this famous mouse model, the agouti mouse model. So these are two genetically identical siblings. They differ only in their epigenomes. And this phenotype is modulated by a mother's diet around conception. So a mother that's fed a diet rich in methyl donor nutrients is more likely to have offspring of the leaner brown variety compared to other mothers who have a diet deficient in methyl donors are all more likely to have the obese golden-colored mouse, which has increased susceptibility to diabetes, obesity, of course, and also some cancers. And this transmission is mediated by differences in DNA methylation at a cryptic promoter of the agouti gene. And this gene is an example of what we call a metastable epiallele. So these are genes that are sensitive to the periconceptual environment, and they have a particular signature, a cross-tissue signature, which is suggesting that the methylation is established very early in gestation, around the time of conception, and is stable thereafter. So for us, if we can identify these in humans, they're a particularly useful tool for us for pinpointing the moment of exposure or the sensitive window of exposure to early gestation around the time of conception. And we, along with our collaborator Rob Waterland in the US, have followed this model in a series of studies now in the Gambia where we've identified human metastable epialleles, largely in American multi-tissue screens. And we've then looked at these in Gambian infants and children to see whether we can see an effect of seasonality or maternal nutritional biomarkers see an effect at these metastable epialleles in humans. And what we see is a pretty consistent pattern. So here's an example, a recent example, in two Gambian cohorts, different age cohorts, the ENIG cohort of 2-year-olds, emphasis cohort, and older kids, 8 to 9-year-olds. And what we see is this consistent pattern of metastable epialleles of increased methylation in conceptions around this time of the year, both in ENID and in emphasis, this time of the year, July, August, September time, and decreased methylation, this is the methylation minima, at this time of year around the February, March, April time. We see this at metastable epialleles And this does indeed coincide with the peaks of the rainy and dry seasons. So it validates our previous work, which has also suggested this in other cohorts that we've looked at. So there's a pretty strong, robust signal of an association between seasonality and methylation at these loci in the epigenome, which other evidence suggests is being programmed around the time of conception and is stable postnatally. Notice we also see a similar signal at imprinted genes, which is interesting if you think back to the plots I showed you earlier in terms of the dynamics of methylation, because this is a time where imprinting is crucially dependent on maintenance of methylation marks right at the beginning of life, the first few days after conception, where these marks, imprinted genes, need to be maintained. They need to evade this erasure of the marks on the sperm and the oocyte methylomes. And there's recent evidence of imprinted genes, again, of the importance of environmental exposure. So this comes from a recent study looking at imprinting disorders, and this time comparing conceptions that have occurred through assisted reproductive technology such as IVF and comparing them to natural conceptions. And they see there much raised incidence of imprinting disorders in ART conception. So we might speculate that, again, this could be a result of the medium at which the ART is taking place in the first few days before the embryos are implanted back in the mothers. So reinforcing this idea that imprinted genes might be another class of genes which are particularly sensitive to the periconceptional exposures. And coming to the end now, of course, we're also intensely interested in linking all this to phenotypes in our population, and we're engaged in a number of experiments and studies now trying to follow up some of these genes where we see associations with seasonality. And I must, of course, mention this important study, very exciting study, which involves a collaboration with Dr. Giriraj Chandak's group at CCMB in Hyderabad. I think many of you may know him, also with Ranjan. This is the emphasis study where we're following up children who were involved in two preconception multiple micronutrient supplementation studies, the Mumbai Maternal Nutrition Project in India, and a similar study in the Gambia. And we're looking at the effects of, potential effects of those supplements, preconception supplements in children, mid-aged children. And we're also looking at the potential, looking into the effects on a whole range of phenotypes, birth measures, growth, cognitive development, and cardiometabolic risk factors, and so on. So I'll just finish up then with a story on a specific gene, which is one of our most interesting candidates we think. And this is the POMC gene, the pro-opiomelanocortin gene. So this gene produces hormones in the hypothalamus, which we know are involved in appetite regulation through the satiety response. So POMC actually mediates the effects of leptin, and its effects on anorectic function. And we know that individuals with genetic mutations in the POMC gene go on to develop early onset severe obesity. And we've shown, along with our collaborator Peter Kuhnen in Berlin, that methylation of this gene is associated with obesity in children and also in adults. And we know that this particular methylated region that we've studied, it does influence the expression of the POMC gene. And importantly, Peter's also looked at methylation in genes from the neurons in the arcuate nucleus, where POMC exerts its effect on appetite regulation, and sees an association in a small sample here between methylation at this gene and BMI. This is in post-mortem samples. When we look at this same locus in Gambians, we see that methylation at this locus is associated with seasonality and also with a number of methyl donor biomarkers in the mothers. And it also has this characteristic methylation signature of inter-individual variation, but a similar pattern across tissues, suggesting it's a metastable epiallele, so programmed around the time of conception. It also appears to be stable postnatally, at least through childhood and adolescence. So we see this comparing newborn blood spots with methylation taken in 12-year-olds, blood samples taken in 12-year-olds. And this has been validated in another cohort comparing cord blood with methylation at 7 to 9 years. So this mark appears to be established in the early embryo and is stable at least through childhood. So bringing all this together, this presents POMC as a very interesting candidate for a causal relationship mediating the effects of early life nutrition and its influence on body weight in children and through into adults, mediated by epigenetic changes, in this case DNA methylation changes. So I think it's therefore interesting to speculate whether what we're seeing here is potentially an example of a predictive adaptive response. So this has been described as a process of developmental plasticity, which enables an organism to change its developmental trajectory in response to an environmental cue. So in the case of POMC, then, we might speculate that what we see as the increased methylation for conceptions occurring in the Gambian rainy season, which is also the hungry season. This is the environmental cue, which is setting up the developing embryo to predict its postnatal nutritional environment, so to predict an environment of nutritional scarcity. This will then in turn decrease the expression of POMC in the arcuate nucleus, which would lead to a reduced satiety response in anticipation of this low food availability post-natally. So this is the adaptive response. And then where an individual then encounters nutritional plenty or relative nutritional plenty, we might then expect there to be a relative weight gain in that situation. So this is in this case, the maladaptive response from this predictive adaptive response. So our hope is that by better understanding these underlying mechanisms linking, in this case, nutrition to later life health outcomes, but really understanding in some detail the epigenetic mechanisms that are involved, our hope is that in the medium to longer term, we might be able to design nutritional interventions, for example, that will improve lifelong health by correcting a suboptimal maternal and potentially paternal. We have evidence of paternal effects as well, which I haven't described today. Suboptimal metabolomes in the parental metabolome, and so reduce patterns of aberrant methylation in the offspring epigenome. Thank you very much for listening.
Thank you, Matt, for another terrific talk. Some of this is difficult stuff. It's very complicated, but you explained it in a very accessible way. Thank you indeed for doing that. Questions, please. Now, you, sir, you're always the first one. We're going to give someone else a chance first, if we have any other over there.
One of the statements you made was about the younger part of the DNA and methylation. Is it that the younger parts of the DNA have more stable methylations and are more likely to be escapees?
Yeah, so that's very interesting. And the opposite is true, in fact. So the the theory is that these more recent additions to the genome they're often inserted into the genome translocations from viral DNA, which tend to jump about as well. That's why they're called transposons, copy themselves across the genome. They're potentially deleterious in terms of their effects, in terms of disrupting gene regulation, for example. So they're pretty ubiquitously methylated as a potential mechanism to dampen down the effects of transcription from these evolutionary young regions. But the speculation or the hypothesis is that at some places this damping effect is incomplete. So there's some instability. And this can give rise to, and the agouti gene in the mouse is one example of this. So the variable methylation at an insertion very close to the agouti gene is stochastically methylated. So in some instances it's methylated, in some instances it's not. And that is affecting the POMC expression. And the metastable epialleles that we've studied are strongly enriched for proximity to some of these transposable elements, in particular endogenous retroviruses. And we think this is perhaps why we're seeing this increased inter-individual differences in methylation, potentially leading to inter-individual differences in gene expression. And this might be where this is opening the door to, in an adaptive situation, opening the door to responses to environmental cues, influencing gene expression, or in some cases, of course, there could be serious disturbances, an adverse disruption of developmental pathways and maladaptive phenotypes.
Thank you, Matt, for your presentation. I'd like to ask you regarding the Angelman and Prader-Willi syndrome. These are the uniparental gene. Both the genes are coming from father in case of Angelman, and both are coming from mother in case of Prader-Willi. And they have got the same type of trinucleotide repeat. And it is diagnosed early for subsequent pregnancies also, that part. So has it got the system to know it prenatally, whether the Angelman or Prader-Willi can be diagnosed prenatally?
Well, I'm not an imprinting expert. I know so there's a severe imprinting disorders are almost entirely due to genetic mutations. And so that's why they may, as you say, be passed from generation to generation. In terms of diagnosing, I suppose it might be possible to do that using the latest techniques using fetal cells, which are present in the maternal blood, but I'm not aware of anyone having done that. But so if these imprinting disorders were to have an epigenetic basis, which some of them do, then that might be possible, I suppose, to detect in that situation. But I think it's most easily diagnosed through genotyping, because it's usually a result of genetic mutations.
Another thing regarding your obesity, there is the OB gene is discovered, the OB, both the recessives are present, then there is chances of obesity also. And in experimental cases, animals, we have seen that by knocking out the OB gene, the animal becomes very much obese also. Now, I like to ask you how this obesity gene is translated to neuroendocrinology. That is the more productions of the white adipose tissue compared to brown adipose tissue. Because brown tissue is more causing the non-severing thermogenesis.
Can
Because
I-
obesity is the disbalance between the intake and expenditure of the calories.
Can I take that one for you, Matt? The reason I'm volunteering is that I've worked with Professor Sir Steve O'Reilly on phenotyping the very rare cases of human OB defects. So we still only know of, I think it's 13 or maybe 15 by now, cases in the entire world of an OB gene defect. So I don't want us to get waylaid onto this too much at the moment. I'd be happy to tell you more in the lunch break. But this is essentially the leptin gene, and the DBDB case is the leptin receptor. And so the pathway, the important part is that the pathway, it's still active through the hypothalamus and through the regulation of appetite, rather than the brown adipose tissue that you were alluding to. Let's take a question here.
Hello. It was a very, yeah, I'm here. Yes. Yes, here.
Oh,
It was a very interesting presentation, and probably we have solutions rather than or answer to some of the problems which exist. But once you find out that this generation disease has been affected by epigenetics, is there any way that it can be undone, or we have to wait for the generations to come? This is one question. Secondly, this rainy season versus dry season, do you think that taking cue from this, there may be some counseling to such populations, that it is better that you think of family in the rainy season or dry season? So will it help? Or epigenetics can be used in a positive way for single amino acid genetic defects, like in thalassemia and sickle cell disease?
Yeah, Matt, Sridhar here on the dais. Just an extension to the first question. It's the same thing, that every answer has a question, but every question need not have an answer. But I just wanted to know that could you be using a drug like azacitidine or something that can manipulate epigenome? That's what she's trying to find out. So do we have some, since we have the problem at hand, can we think about a solution? That is, and at what time period should it be when do you intervene?
right, yep. So these are really excellent questions. So I think the answer is complex, and it depends on what gene you're talking about and when the gene might be most sensitive. This is one part of the answer. So, for example, the genes that appear to be programmed periconceptionally, then one answer would be to optimize the diets, ensure that we optimize the diets of the mothers periconceptionally. But also many of these genes may be another sensitive period. Some of these genes can appear to be able to be reprogrammed later, postnatally, particularly so in the fathers prior to conception of the next generation, and in the mothers during adolescence. So that might be another area where we might be able to propose dietary interventions. But I think there are other possibilities as well. So I think there are opportunities potentially in personalized medicine, for example. So taking the POMC gene as one example, and we've been discussing this with other potential collaborators. So we know, for example, when we look at using bariatric surgery to treat severe obesity, that some patients respond very badly. Some patients respond very well to bariatric surgery, and some respond very badly. And factors like the methylation, epigenetic modifications in the POMC gene might play a role there, because if the POMC gene is blocking the satiety response, then we might suppose that bariatric surgery would really have no utility in treating obesity. So that's another part of the answer. And another part of the answer might lie in the future in terms of technologies which are being developed, sort of CRISPR-based technologies, where we might actually be able to target specific genes and specific epigenetic modifications and actually correct them, go in on the precision genomic level and correct them. And those are being developed right now.
Yeah, because it's more of an interplay of leptin, ghrelin, gastrin, the microbiota. There are so many in the picture. So just giving so much of importance to just one POMC gene and making it very simple. You just feel that if I have an agent that could work on acetylation, methylation, or histone, whatever is contributing to the obesogenic phenotype, I could have an answer for it. That will be so easy.
So our collaborator is working on these. The problem is the issue of targeting that Matt just alluded to, because although there are compounds that will alter methylation, they tend to be very broad spectrum, so you'll be changing all sorts of things. So the issue, and you probably, I mean, I'm sure you're well ahead of me on this, but there's a lot of interest in pharma in relation to such compounds, but it's the targeting that's the issue. Yeah. Lynette.
So first of all, very grateful for a very accessible presentation for those of us who don't live in the world of genetics. But for that same reason, when I hear this discussion about the possibilities of addressing this challenge, to me, the first thing we should be thinking about is addressing the underlying food insecurity that is clearly happening in a part of the season that you're seeing there. And I think you built that story very nicely. You showed the differences of intake, well, not intake, but I've forgotten what it was, the nutrients related to the one carbon cycle. You showed how various those are between the rainy and the dry season. If we deal with that, how much do you still need all of these more complex clinical approaches to addressing this are we going to need? Or can we actually deal with this by having a proper, diverse diet from the beginning?
Yeah, I guess the answer is we don't know yet, but neither do we know what the specific nutrients or nutritional factors are that are key. And one thing. So, for example, when we started these studies, we expected that we would see the opposite effects that we expected, because the rainy season is in a sense the nutritionally challenged season, where the previous year's harvest is depleted, and that's why it's known as the hungry season. So calorific intake is much lower then. But it turns out that we think, well, this is much more a story about micronutrients in terms of methylation, which perhaps in hindsight should be obvious because of the one carbon pathways. But we still don't know which particular nutrients might be important. And when we're looking at this in some detail now, and it seems that the particular nutrients, in some of the modeling we've been doing, the particular nutrients which might be driving DNA methylation actually vary across the seasons. So there's an interaction between seasonality and the important nutrients. So this is suggesting that it's the nutritional background, the background nutritional status is itself important, which suggests that these are going to be very population specific, potentially, any interventions that we might try, which obviously adds a whole other layer of complexity here.
Final question.
We're talking about a cycle between a rainy and a dry season, was one of the things that happened. Is it possible that this cycling between poverty and plenty kind of thing is more likely to cause methylation than having a stable environment?
Yeah, I mean, we have to remain agnostic on what are the seasonal factors driving this, because they're all interrelated. We see the seasonal variation in nutrition, but there are also big changes in BMI of mothers across the season. And it's very hard to unpick these. And all of them have been identified in other populations as factors which can influence methylation. So this is an ongoing project. And one of the ways we tackle this, of course, is by designing interventions to see at some stage what it is that will affect the methylome. But absolutely, there are many other factors that vary seasonality, including toxins such as aflatoxin, which could be playing a role, pesticides, so on.
Matt, thank you very much indeed again for making a very difficult topic accessible. Thank you.